end of the time. So few specific hopes that we have bringing foundation models to the field. Okay? One of the gaps that we have in the field is we know that there is a huge amount of animal model data about the brain, right? Or cellular level data about the brain. Those who work with Allen Brain Atlas, those who work with all the animal or non-human primate atlases that we have very, very interesting data inside, that we have never been able to use it very much for human brain. And then we have huge amount of human imaging data, right? So, but there's a gap, how these things are going to be related to each other. So these multi-dimensions of brain data that we have, from cell level to basic brain circuits, to non-human primates, animal models, to human databases. AI showed that it can basically integrate and do what we call multimodal data fusion. And being able to use those models to develop things like cross-species foundation models. And developing a specific joint spectral embedding between different networks across different species, which is mind-blowing in terms of how those things are going to work together and how they are going to integrate different levels of data together. So the new things that are happening in the field is just unbelievable. And then in the field of brain imaging and using multimodal brain imaging for brain stimulation, those who are doing fMRI, they know that fMRI is doing something, but it's not covering everything, right? And those who are doing EEG, they always say, oh, EEG is much better because EEG can do this, you cannot do that, let's just stick with fMRI. But how we can bring them together? How that we can have a model that can read both MEG, fMRI, DTI, putting them all together in one single data, and then have both kind of spatial and also temporal characteristics together in one single model. And this is what we have as fusion pipelines, to combine the anatomical scaffolds to oscillatory states, to dynamics that we have with both, with the conductivity models that we have, or even adding the computational head models that we have with brain stimulation. And then adding, I'm not sure if John Griffin is here, so John is sitting on the back just making notes to find errors in my slides at the end of the talk. But yeah, John is developing lots of fantastic models in cellular levels, and how those things, John, could be integrated with these models together, and how things that Z was mentioning, that we cannot differentiate between 5 Hz stimulation and 10 Hz stimulation, right? But John can do that with his models, or even differentiating between things like ITBS and different patterns of ITBS. Those models, how they can come together in a single. Sounds like a dream, but it seems that it is moving forward for that specific dream. And using those foundation models for downstream tasks, for specific closed-loop stimulation, personal perceptual stimulation, composite biomarkers that would have data from different levels of databases that we are collecting together. And these figures are from a new paper that I will show you at the end, that I will share the preprint of the paper. So it is already submitted, so I will share the whole paper that will include all those figures that are coming from that specific paper that we put together. So I will share the data in that paper.